This
Cancer Research.
conference will now be recorded.
Ma'am has also received Best Doctor Award from Tamil Nadu State Government and was awarded the Best Paper for Mesenchymal Stem Cells for Acute Liver Injury, a preclinical study at 16th International Hepatopancreatobiliary Conference at Jaipur. We welcome you, ma'am.
Yes. So thank you, Akash Pandit. Thank you for this intro for the chief guest. And so we have just been knowing like for her awards and recognitions she has received from the government and from other funding agency. We wish you very all the best, Sekunda. Now our audience is there waiting eagerly for to know about the research opportunities in life sciences. As I was telling you in the introductory session, students, a multidisciplinary, interdisciplinary research is always highly essential in today's world. Maybe a CSE engineer or a ECE or a mechanical civil AI is used so much in medicine. I was telling you about the nano robots in surgery. And so there are so many, like, without software, there is again no all our X-ray imaging. In my very first session for the physics in life, I was telling you about how physics was playing a major role in this pandemic also. I said the coronavirus structure is being imaged by a physicist only, so he could image the structure and give us a structure with the spikes. So every field is useful, is being no intervening and penetrating and overlapping in any other fields, and we are able to bring about the real research in our country. Now here we have Dr. Sekunda to explain about the life sciences research for us. This would be a generic session for all of us, and I'm sure it's going to be very interesting. So yes, Sekunda, now the session is yours.
Thank you, Sureka and Akash, for the very nice introduction. First of all, I should thank Sureka for the opportunity to interact and share my thoughts on this topic with young minds like you. So, Sureka, how shall I share my screen?
Yes, Sekunda.
Are you able to see, Sureka?
One sec. Students, are you able to view?
Can you see my
Yes, ma'am.
screen, students?
Yes, ma'am.
Yes. Okay.
Yes.
Okay. Thank you. That's fine. First, I should apologize for the delay. I think it was due to a technical snag on my side. So I'll just, since we have very few minutes left, I think, can I take 20 minutes or so, Sureka? 10:45?
Yeah.
Is it okay?
No, you can take till 10:50 also.
Yeah.
No problem.
Okay.
Yeah,
Okay.
because the next
thank you
session
so
is
much.
going to be our alumni, our student only. So I could tell him, so you can take your session.
Okay.
Still 10:55, you can go on. Okay. Yeah.
Yeah. Okay. Thank you so much. So first, I would like to congratulate all the freshers who have chosen this field as your career. It is very, very promising. So I will tell you what all promises this holds for you. I'm sure you would have searched widely before taking the decision of choosing a life science subject. Okay. So what are the recent innovations in life sciences? So what does life sciences have for you? It presents many opportunities. So in my talk, I will be telling you what are the various opportunities that are open for you students. And one thing very interesting about this field is it is constantly evolving. Okay. So every day there is going to be a new advancement. It's a growing field. Next, there is going to, as Sureka rightly pointed out, only a multidisciplinary and an integration will be useful for us to move forward. So integration of life sciences and technology will be the key for addressing a multitude of pressing global issues in healthcare settings as well as beyond. So let's see what were the innovation in science over the last century. So if you see in the year 1928, first antibiotics were discovered. So we all know how life will be without antibiotic. So no surgeries can be performed. We all will die of infections if there was no antibiotic. So this discovery of antibiotic was a major milestone in history. Okay, so we know that we treat all bacterial infections from your typhoid, your typhoid, your cholera, what else and what not. So many diseases, your bacterial diarrheas, your streptococcal throat infection, everything is treated using antibiotics. Next, there was discovery of this gel electrophoresis. So you have both DNA as well as protein gel electrophoresis. So this helps in your genomic as well as your proteomics study. Next, in 1951, there was HeLa cell discovery. So what are HeLa cells? These are used in tissue culture. So you can cultivate viruses in the cell lines. Okay, you can propagate the virus and you can use it for various studies towards the diagnosis of viral infections, as well as you can use it for many other things. Okay, then the structure of DNA was studied in the year 1952 to 53. So you all know who discovered the DNA, structure of DNA. So
Robert
Watson and
Brown.
Crick. Okay, then you have the DNA polymerase enzyme. So DNA polymerase enzyme is used in polymerase chain reaction. Then you have the reverse transcriptase enzyme. The whole world is talking about this COVID pandemic, and we all know what was the test for diagnosing SARS-CoV-2 infection. What was the test? Yes, it was the real-time PCR. Okay, so without COVID-19 RT-PCR test, you cannot go anywhere abroad. You need to show a negative report. And whenever you suffer from a
Sekunda,
viral, yeah,
Sekunda, sorry for interrupting. Students are saying we're not able to
yeah,
see
tell
your
me.
screen. Your screen is not visible. Can you give the share option
Oh,
again?
okay, I'll do that. I'll do that. Initially, you were able to see, no? Share. Now, Sureka? Sureka, are you able to see my screen?
No, no, it's not still visible. Students, are able to see the screen?
Ma'am, I kindly to give the share option, ma'am.
Yeah, now it is coming. It is coming. Yes. It is getting
Is
loaded,
it?
Sekunda. Yeah, it's getting loaded.
Okay.
Uh,
I'll just continue talking.
yeah.
Or you want me to?
Students, you're able to see the screen now?
No, ma'am.
No, it's still in the loading only, actually. Yeah, now we could
But I
see.
gave the.
We could see now. Yes,
Okay.
we could see the screen
Yeah.
now. Yes, you can continue
So
talking,
we
please.
have this, yeah, the real-time PCR. So for doing real-time PCR, this reverse transcriptase enzyme is very, very important, which was discovered in 1970. Then you have a lot of these restriction enzymes for digesting the DNA fragments. Then you have PCR, which was discovered by Carey Mullis in 1983. So biotechnology students, your bread and butter is PCR. Then you have bioluminescent markers. So for doing in vivo imaging, these markers play a key role. Then gene therapy. In my talk, I will be discussing about what is gene therapy. Then you have this RNA, CRISPR, whatnot, and okay, so since all these innovations happened over the last century only, in this pandemic, we were able to fight against the SARS-CoV-2 virus. If not for all these technologies, you couldn't have imagined a diagnostic tool being available to the population all over the world just in a matter of a month's time. And also about this vaccines. See, it is a really a wonderful thing that we now have vaccine for COVID. Otherwise, just imagine the mortality we would be facing. See, already in this pandemic, many of them have lost their kith and kins. If not for the vaccine, the catastrophe would have been much more. So, thanks to the innovation in sciences which helped us to face this pandemic. So now, what is the key for a life science innovation? When will it be beneficial? So any innovation should think of what is the target. Like it should satisfy what the market needs. That is what the society needs, what the population of interest needs. So it should be translational. Okay. Always research can be classified into two: basic research and translational research. Okay, they are two sides of a coin. You cannot separate them. Without basic research, there is no translational research, and without translational research, there is no use of doing a basic research. So always research should be focused in these two angles. You should have the basic research and translational research complementing each other. What is very, very important in research is that always many forget the ethics behind it. It is very, very essential and important that your research is ethically done. Okay, only whenever it is ethical, it will stay the test of time. Otherwise, your victory will be very short lived. Okay, so whenever it is ethical, though it may be slow, but your target will, whatever you have achieved, will stand the test of times. Then already I told you, it has to have the ability to be translated, as well as sometimes it will go in for commercialization. Then you have to produce a safe and effective product that will be useful to the masses. Last but not the least, price. Pricing is very, very important. See, the common man should be able to afford the product that you are producing. If it is going to cost heaven and earth, then there will be very few takers for your product. Say, for example, the COVID vaccine. See, it was available free of cost in the government sector for a very large population, and for few affordable, they took their vaccination in the private sector. The cost was also pretty low. Okay, so whatever product you produce should be, the pricing of the product should be acceptable for your routine clinical practice. Then what else should you think? That is, you should always work along with nature. You should not go against nature. Then you should nurture your thoughts and ideas. See, you may be having very many wonderful ideas in your mind, innovative minds. Okay, but you cannot just stop with thinking. Okay, you should put your ideas into practice. So how can that be done? By scientific expertise. So you have to research. Okay, research can be just reading what is there already available in the literature, then open up and see how you can make your research work novel. Okay, so for that you need scientific expertise. Next, you need support from your institution, as well as the infrastructure required to do your research should be available. Then you should be willing up to join up with people, collaborate with various institutes, with common research interest. You should be able to collaborate with them. And last but not the least, you need funding for all these. So what are the probable barriers to innovation? So the first thing that comes here will be the funding. So in India, if you see, there are various funding agencies like Indian Council of Medical Research, which funds for medical and health research. And then you have Department of Biotechnology and Department of Science and Technology, which are funding for your biotech research. So there are many central government institutes that fund for your research, as well as there are also international funding agencies. Another important thing is the time. Okay, so you cannot do a research overnight. Okay, it will take a lot of time. Lot of patience is necessary for you to progress in research. Then the complexity. Research is always complex. Okay, so it needs a multidisciplinary team approach. That is the key for doing any research. And then whatever topic you choose should be academically very demanding. Okay, it should be of interest to many. Then only your whatever innovation you are doing would be applicable to the society. Then you have a lot of, once you have identified a product or you have developed a product, there will be lot of patenting issues. Like people in the IIT and such institutes, they are familiar with this patenting issues. So we can take their help and go about getting patents. Then there should always be a linkage between academic institution and industrial firms. Okay, so that will be helpful in taking your innovative research to the market. Then next, I already told you the difference between basic science versus translation. So in medical terminologies, what we usually say is from bench side to bedside. Okay, so bench side is the work that we do inside the lab, bench side. So taking it to the bedside, bedside is the patient. Okay, the patient is admitted in the hospital. So taking the bench side research to the bedside, the patient on the bedside. So these are, there are barriers, I would agree, but there are also methods how we overcome these probable barriers. So what are the, so because I am a medical graduate, more so I will be discussing about the recent innovations in the healthcare sector. So that includes genome editing or gene editing. Then you have personalized medicine, then regenerative organ development, personalized diagnosis, development and production of drugs and vaccines. Now we will go on to one by one. So what is gene editing? Gene editing is nothing but process of making a tiny control change in the DNA of a living organism to get a, to produce a genetically modified organism. So this gene editing technology has been done all over the world, but there are lot of regulatory bodies and regulations that control this gene editing process. Okay, we have to be very, very clear. Whatever I already told you, whatever we are doing should be very ethical. Okay, and we should follow all regulatory guidelines when we pursue such research. So what do they do in genome editing? So they remove the cells from our body. Okay, and then they isolate the DNA. After isolating the DNA, they do correction, genetic correction. Whatever they don't want, the unwanted fragments will be nicked or they want to introduce some other fragment, beneficial DNA fragment, then that will be introduced. And then these corrected cells will be cultured in a petri dish. After culturing, they will do transplantation of this. What is autologous? Autologous means that from this patient I take the cell, I inject it into the same patient. Okay, so that is autologous transplantation. So these corrected cells, by means of certain viral or non-viral vectors, injected into the patient's body. So this is called as genome editing. So what are the opportunities and challenges of this gene editing technology? So opportunities is you can do targeted medicine, then you have the opportunity of producing biofuels, then you can get even disease resistant crops, and then very many new industrial products like vaccines. Okay, many products are obtained by recombinant DNA technology. What are the challenges of this gene editing is the safety. Okay, so first it should be very safe. Then you have ethical concerns. I always stress the importance of ethics. And then there is a lot of regulatory challenges. Anywhere you go, in India, abroad, everywhere there will be lot of regulatory concerns. Okay, it's all for our good only. In India, we have the Drug Controller General of India, DCGI. So you have to get approval from DCGI before you produce any product for therapeutic use. In US, it is called as FDA. Okay, so these are the regulatory agencies. Next, going to gene therapy. So gene therapy is somewhat similar to gene editing. So what do you do in this gene therapy is you correct a mutant gene by expressing a healthy copy of a gene. Okay, either you can introduce the healthy copy of a gene into the genome, or what you can do is you can inactivate or silence the mutant gene that is responsible for the disease per se. Okay, so this is done using the gene editing tools which we discussed earlier. Okay, so that's about gene therapy. So gene therapy for hemophilia and many such diseases are under research. Okay, so that completes about gene editing and gene therapy. Next, going on to digitalization of healthcare system. So this has become the key nowadays. So what is that? It is wireless medical sensor network. Okay, so previously, if you have to take a temperature of a patient, what we usually do, you put it inside the mouth or you put it inside the axilla, or for kids in the rectum, all that stuff, creating a lot of discomfort to the patient. But now, what is the trend? You might have seen it everywhere in your day to day, wherever you go, in the metro stations, in the malls, everywhere. What do they do? How do they check your temperature? Yes. So they use a sensor based thermometer, so that doesn't cause any discomfort to us. So it is very easy. And there are your pulse oximeter, then you have a digital BP apparatus, what not. So everything is digitalized for the patient comfort. So we have very many handheld devices. So all this is a result of what? It is a result of the innovations that has been happening in the last century. Okay, so this is the revolution in digital healthcare. Next, going on to what is personalized medicine. Personalized medicine is nothing but tailoring the medical treatment to the individual characteristic of each patient. To make it simple, I'll give you an example. There are three persons, X, Y, and Z, each having a temperature of 100 degree Fahrenheit. They are suffering from fever. Okay. For the patient X, I'm going to give him 500 milligram of paracetamol. I give him Crocin 500 and he responds well. Okay. Next, I'm giving same Crocin 500 to Y and he does not respond. So what I do is I increase the dose. I make it Dolo 650. Then that patient responds. Okay. Next, there is Z. Z doesn't respond to paracetamol itself. Though all the three are having the same temperature, X is responding differently, Y is responding differently, Z is responding differently. So to treat Z, what I am doing is instead of Dolo 650 or Crocin 500, I am giving him 0-Dol P, which is aceclofenac. Okay, so he is responding only to aceclofenac. Okay, so what you should understand is each patient's body responds to the same drug differently in a given similar situation. Okay, but because we do not have personalized medicine, so we do generalize the dosing of medicines and we are giving. Once this personalized medicine becomes available, I will know that X requires 500 milligram, Y requires 650 milligram. For Z, I should not give paracetamol. Instead, I should give aceclofenac. So that is personalized medicine. Okay, so I think you can understand what I'm trying to say. Then how do I find out how X is going to respond, how Y is going to respond, why Z is going to respond? So for that, you have to, how you can find out. See, it is mostly used in cancer research. What they do is they will take the blood sample, okay, blood or some tissue from the cancer patient, try to isolate the DNA or try to isolate the cells from the patient, allow it to grow on a petri dish, then subject it to various concentration of the drug and check at which concentration these cells respond to the medication. So based on your research output, you treat the patient with that dosage of drug. So this is how personalized medicine plays a role. Next, going on to nanomedicine. Okay, so we all know we are in meters. Okay, we are in, our length is in meters. Then if you take small tissues, it will be in millimeters. If we talk about cells, we know they are several micrometers in size. Then you have what? You have molecules. Okay, small molecules within the cells. Their diameter will be in nanometer. So the medicine that deals with the study of these molecules is called as nanomedicine. Okay, nanomedicine is a highly interdisciplinary field that requires the researchers to have a very strong understanding of the nanomaterials and how they interact with the human body. Okay, so this nanomedicine is useful in drug delivery, gene delivery, for detection and diagnostics, molecular imaging, targeted therapy, and biomarker mapping. So this is how nanomedicine has been used in orthopedics department. They do bone tissue engineering. Then you have lot of nanostructured implantable materials. Then you can modify the surface and coatings so that the tissue graft takes
என்ன சார் ஆச்சு பாத்தீங்களா
biomaterials for regenerative medicine. So I quickly go through. I think time's up, Sureka.
No, you can
Shall I
continue.
close?
Another 5 minutes. Last 5 minutes.
Okay. So I'll quickly go through what is regenerative medicine. So we all are hearing a lot of news about stem cells. Okay, so stem cells, tissue engineering, and biomaterials put together is a field.
Sekuntai, sorry for interrupting. You're able to hear me? Your voice became inaudible actually. Yes, yes, it's inaudible, students. One second, I'll just check.
So undifferentiated means, what is undifferentiated? That is, it doesn't have a property of a particular cell. See, if you take a muscle cell, it will behave like a muscle. Okay. If you take a cell from the skin, it will behave like a skin cell. But if you take the stem cell, it will not behave like any cell. But you can coax it to become any other cell of your interest. Okay, so that is stem cell basically. Then what are these bioactive factors? Bioactive factors are nothing but the cytokines, the growth factors, hormones, and other morphogenic proteins that help for the growth of these stem cells, for the growth and engraftment of these cells onto scaffolds. What are scaffolds? Scaffolds are nothing but the structural support or the extracellular matrix that will hold these cells. For example, cells are like the bricks, and this scaffold is like the cement holding the cells together. Okay, so that is regenerative medicine, and it is a very, very promising and an upcoming field. And so far, there has been the hematopoietic stem cell transplantation is already approved and is in use for patient therapy. Treatment of leukemias is done using this bone marrow stem cells. And now for critical limb ischemia, certain products in the market is available and has got DCGI approval. And there are many other applications of stem cells which are still in the clinical trial stage, which are in the research stage, and it is evolving, and let's hope it has a lot of promise in the future. Okay, so this tissue engineering is somewhat similar to regenerative medicine. Here, the scaffolds and the decellularized organs play a vital role. So there is something called 3D printing. Okay, 3D printing is nothing but you can print the structural components. Like for example, for somebody the knee joint is collapsed, then you can print the cartilage of the knee joint using cells and the scaffold in an organ printer. That is called 3D printing. So that is available. So tissue engineering holds a lot of promise. So these are the biomaterials available. Okay, so I already told you, for replacements, for heart valves, for dental implants, for contact lenses, for cochlear implants, many other things, these biomaterials are useful. So what they do is, first they will grow the cells in culture, then allow a sheet formation for the extracellular matrix formation. Then they will decellularize the ECM and use this ECM sheets, the extracellular matrix sheets, to grow some other cells of our interest. Okay, so this is about biomaterials. Last but not the least, I just will touch fastly upon artificial intelligence. I heard in the introductory speech Sureka talking a lot about this artificial intelligence. So it's a branch of computer science which emphasizes in building smart machines capable of performing tasks that typically require human intelligence. So it is artificial human intelligence. So machines with human intelligence is artificial intelligence. So it is gaining popularity and it is influencing the way we live, interact, and improve customer experience. So, types of artificial, there are reactive machines, theories of mind, limited memory, self aware artificial intelligence. All these are the various types. And what are the technologies that make use of artificial intelligences? You already know Siri, Alexa, all that. Then you have lot of self driving cars, Tesla, then you have robo advisors, then email spam filters, flying drones. All this use artificial intelligence. What is the application of AI in healthcare is for doing medical image analysis. It's useful in drug discovery. In many clinical trials, they are using that. Then for medical diagnostics. And we all know about artificial intelligence complementing the robotic surgery. Then last, we are discussing about robotics in healthcare. So you have these technologies which these robots are helpful for the surgeons to perform surgeries. Okay, so all these innovations are very much beneficial to the medical field. Okay, so we have touched what are the various things. You have regenerative medicine, you have personalized medicine, you have gene therapy, then production of vaccines and drugs, then overall it improves the healthcare system. Thank you for your patient listening and sorry for the delay initially. Thank you.
Thank you, Sekunda. Thank you for this noteworthy, generic, about the drugs, about the personalized medicine. Today we have gained knowledge. I'm sure all my students would have got the knowledge on the biomaterials, because this biomaterials you all will be studying in your second semester material science in engineering physics itself. And this is useful for all. These are materials, like without materials we are not, there is no human life. We are always dependent on materials. And the usage of biomaterials, I think once again we will ask Dr. Sekunda to come to us physically after the class is resumed. So we want you one day to come and give a talk on this nano research in detail, because it will be useful for the biotechnology students. And our next chief guest is
Definitely.
actually our final year ECE student. He was saying about the augmented reality surgery also. So when you were mentioning about the AI, he was just typing in the chat box. And
Okay.
sure, students are, thank you so much for touching upon this AI. For them it will be like very interesting also. Thanks a very lot, Sekunda, for dedicating your time and energy for us for the couple of minutes to share the very wider ocean of knowledge. In that, a little drop is what you shared. I know. So again, in another occasion we want you back here in our college physically. And I'm really feeling really blissful and so blessed to have you today here. Thanks a very lot, Sekunda. Any questions? I will just. There are so many students posting
Yeah,
thank you
sure.
there in the chat box. There are so many students saying, yeah, informative and wonderful session. So many of our teachers were also there. All my first year staff members were also here. Chemistry, maths, English, and physics teachers. Yes. So students, any doubts you can actually now be open. If you have any query, you can just ask now. Yes, Lakshmi Narayana. Lakshmi Narayana says wonderful session, madam. So very, yeah, thank you. So any down students? Yeah, Bhargav is asking, ma'am, what is your opinion on 3D printed organs? Are they safe?
3D printed organs. See, when you use autologous cells in those organ printing, then it is safe. It is safe only in certain areas. You cannot see the heart valve already. It is in use actually. The Triple M, no, Madras Medical Mission. There they are doing these heart valves. Okay, from the patient's own stem cells using the valve scaffold, they are making artificial valves, biomaterial, engineered valves. So it is safe. Like you cannot use it for every organ. Okay, so at specific areas it is useful. Now, so far, what has been proven is heart valves, and then for joint prosthesis, there it is safe. But others are all investigational. Like they have tried to create an artificial bladder in the lab, then they have created an artificial trachea in the lab, all that. So all that is in only research stage. Okay, so very nice of the student to have thought of the safety. That is very important. Safety and ethics is
Yeah, he
what
is
I
our
would
final year
always
student.
give importance. Yes, safety and ethics is very important aspect of any research.
Yes.
Have
Any
I
other questions,
answered your
students?
question?
Yeah, he has replied saying that thank you, ma'am. Got on clear image. That's what he has replied to you,
Okay.
Sekuntha. Yeah.
Okay.
The next question, yeah, ma'am, when you're available, share the role of AI in medical in brief. So yeah,
Sure,
Anirudh,
I'll
I
do that.
think probably this recording. Yeah, once she shares to me, I will just post in your WhatsApp group. Okay, so you can again have it. So she will be sending me. Pranavi
Okay.
says thank you,
So
ma'am, for your
you want my
presentation. Yes.
PPT, right? You want my PPT, right? I'll share it with you, Sureka. I'll share it to your
Yeah,
mail
okay,
ID.
Sekunda, so that I can post in the. Sure, thank you.
Yeah.
So I'll post it to the students.
Yeah, sure. I can do
So
that.
yeah,
I can do that.
Srinivasan sir, you're there. Yeah,
Yes,
Srinivasan
I'm
sir.
here,
Yes, sir.
ma'am. I'm
Yes, sir.
here.
Yes, sir.
Am I audible?
Yes, sir, you're audible.
Okay. First of all, I like to thank you, ma'am, for the wonderful presentation. That's one thing. And apart from that, I don't have any. I don't got any direct contact to thank every doctors. So in this pandemic, really, you are doing a wonderful job, and even we are surprised that what the doctors are doing, because they are seeing one patient and the other patient, so continuously they are seeing each and every patient. So I take this occasion to thank all the doctors on behalf of all my faculty members also, even though it's not a right platform, but this is the great platform, I think so. And second thing about this seminar, uh, uh, I didn't know that the whatever the medicines the doctors are prescribing are very personalized. So as you said, aceclofenac or paracetamol, that will not respond to everyone. So that is one of the knowledge I got today. And another thing is, in the case of regenerative medicine, I always fond and surprised about how they are printing all the organs with the 3D. And I think, as you said, the heart valve has been printed already in Chennai itself. That is really surprised to hear that. So I didn't know that the real time the people are using the 3D printing models uh, for the organs, whatever the, because we have to always go for the donors in case of any kind of
Yes.
failures. So that is also another new thing, and it is also difficult to get donors. I also understand that.
Yes.
And so these are all the new things. And I think if you got more time, maybe you might touch upon the AI and all those robotic surgeries and all. Students will be so much interesting in that thing, I think so. I could able to see in the chat box
Definitely.
that robotic surgeries, they are all very interested, I think so. So you have to give another session, ma'am, for this robotic surgeries and all those things whenever you are getting time. And thank you so
Sure,
much
sir,
for
sure.
this wonderful session, ma'am. Thank you.
Thank you, sir, for the nice words. Thank you.
Thank you, ma'am.
Yes, thank you, Srinivasan sir. Thank you so much.
Thank you,
And
ma'am.
yeah, so it was again our chemistry staff has said that it was a very wonderful and informative session. Yes, because chemistry staffs will be knowing more about this drugs. Okay, so they're also like feeling so thankful to you. Thanks a lot, Sekuntam. Again, we wish you very all the best, as I said in the beginning. Again, we'll meet in some other occasion. Thanks a very lot.
Definitely. My pleasure. Thank
Yeah,
you. Thank you, Sureka. Thanks for the
thank you.
opportunity.
Yeah.